Hello and welcome to the Clinical Care Options podcast series, a closer look at evolving treatment strategies for rheumatoid arthritis. Today's episode, taken from a recent symposium, is on JAK inhibitor efficacy and safety. We'll hear from Dr. Stanley Cohen, Dr. Sheetal Desai, and Dr. Ruderman as they interpret the latest data for JAK inhibitors in RA. It includes the oral, Beacon, Beam, Select, and FINCH series. Also included is an in-depth discussion of JAK safety, including MACE and venous thromboembolism. If you want to follow along with the slides, just go to the show notes and click on the link. You can also find a link to additional online education on RA from Clinical Care Options. So let's get started and hear about JAK inhibitors in the clinic. We'll start with Dr. Stanley Cohen.
So in the next few minutes, we're going to talk about the clinical data that led to the approval of the JAK inhibitors. So all of the JAK inhibitors for registration with the various agencies around the world had a very similar development program. They studied these medications in three different populations: biologic DMARD incomplete responders, methotrexate incomplete responders, and methotrexate naive patients. And they all had different acronyms, which some of my colleagues can remember very well, and for me, it becomes a word salad. But again, the studies were all designed in a very similar way to get approval from the regulatory agencies. And Eric mentioned the other kinase inhibitors, the MAP kinase inhibitors, the SYK inhibitors. We spent over a decade looking for small molecules, oral molecules, that would be as effective as biologics, and had no luck until the Janus kinases came into evaluation. So these are the various phase 3 studies for the biologic DMARD incomplete responder RA patients. And these studies are shown here. I'm not going to read these in detail. They were 24 to 52-week studies. The primary endpoints were generally at week 12 and were ACR 20 response, or if for EMA approval, DAS28 CRP less than 3.2. They generally were large studies. They were placebo-controlled, generally with patients on background methotrexate or conventional synthetic DMARDs. And for several of the studies, they were dose ranging, tofacitinib, the 5 milligram BID dose, which is the approved dose, and the 10 milligram BID dose, which was evaluated, which was not subsequently approved. For baricitinib, it was 2 or 4 milligrams, down from 8 milligrams in the phase 2 program. Upadacitinib, 15 milligrams in one study in comparison to abatacept and the Select Choice, which will be presented at this meeting, was presented at EULAR. And then the 30 milligram dose in the Select Beyond study. Filgotinib moved forward with 2 doses, the 200 milligram plus methotrexate, compared to patients continuing methotrexate with placebo. So we'll run through this data quickly, because the data, what it shows in these studies for biologic DMARD failures was that it works. For all of the particular JAK inhibitors, this was the first trial with tofacitinib plus methotrexate. And you can see here on the graph that there was superior efficacy measured looking at the ACR 20 response at week 12 for both doses of tofacitinib plus methotrexate compared to continuing placebo. This was at 12 weeks, the primary endpoint. For baricitinib plus methotrexate in TNF-IR patients, 527 patients, 2/3 had failed two or more biologic DMARDs. Again, looking at the graph, like ACR 20, ACR 50, and ACR 70. The light blue is the 2 milligram dose. The darker blue is 4 milligram dose of placebo in orange. And again, you can see statistically superior efficacy for the JAK inhibitors. And as we saw with our other biologics, in the TNF-IR or biologic IR population, the ACR responses are not as robust as they are in earlier disease in patients who are methotrexate incomplete responders. The Select Beyond study was, again, looking at upadacitinib plus methotrexate in the same population. On my left part of the slide, ACR 20 response, primary endpoint week 12. You can see that for both doses had a similar ACR response of about 60%, superior statistically to about a 24-25% response with placebo. And after re-randomization, the placebo patients who were re-randomized to upadacitinib 15 or 30 milligrams nearly caught up to the original cohort. And then on the right part of the slide, the DAS28 CRP less than equal to 3.2, low disease activity. You can see, again, 42 to 43% of patients, which is really a very large number for a biologic DMARD incomplete responder population compared to 14% on placebo. Some of this may be due to the impact of IL-6 and reduction of CRP, but very robust response. The select choice, which again is going to be presented at this meeting, was also presented at ULAR, was upadacitinib plus methotrexate and biologic bDMARD-IR responders. It was one of our first head-to-head comparison with abatacept, and they had a non-inferiority design. And on the left part of the slide, you can see that upadacitinib at the week 12 primary endpoint was not only non-inferior, but superior to abatacept plus methotrexate. And then when you look at the ACR responses, you can see at week 12 that abatacept in orange and upadacitinib in the bluish color was statistically superior for ACR 20, 50, and 70 at week 12. Now, week 24, abatacept starts catching up. So if you want to design a clinical trial, you put a little quirks in there so you're going to try to win your trial. And that's what they did by an early primary endpoint. But clearly, upadacitinib was effective in this group of patients and somewhat better early on than abatacept plus methotrexate. Now, filgotinib, also similar trial design, taking a look at patients on background conventional synthetic DMARDs, primarily methotrexate. And what you're looking here on the graph is the ACR 20 response at week 12 primary endpoint. And both the 200 and 100 milligram were superior to the patients continuing conventional synthetic DMARDs with placebo. So what we're showing you here is just the ACR responses or DAS28-CRP. As Dr. Strand mentioned today, these therapies are quite effective at patient-reported outcomes. Radiographic outcomes in general were not looked at in the majority of these biologic DMARD failures, but were looked at in the methotrexate incomplete responders, and that will show you that data. So this is one of those slides that you usually see in your ophthalmologist's office. But this is, again, summarizing the studies that were done. So multiple studies were done in methotrexate-IR patients for all of the approved and the EMA-approved JAK inhibitors. In general, this was the population where they compared their JAK inhibitor to a biologic, and adalimumab was the one that was chosen by all of these JAK inhibitor developers. These were the studies where radiographic outcome was looked at, such as the oral scan with tofacitinib. It was also looked at in RA beam with baricitinib. So we'll highlight you this data. These were generally 6 months to 52 weeks studies, large populations of patients with dose ranging studies, looking at radiographic outcomes. Most of these were dose ranging, as I mentioned. You can see on the bottom column, the ACR 20 response was generally the primary endpoint, although in a phase 3b/4 study, the oral strategy, the ACR 50 at 6 months was the primary endpoint. Okay, so here we're showing, again, same type of depiction we showed you with the biologic DMARD failures. This is methotrexate incomplete responders and 2 trials with tofacitinib, and have the placebo in orange, the 5 and 10 milligram BID dose, and the adalimumab comparator, which was just an active comparator in this study. And you can see that the clinical efficacy for the ACR 20 at 6 months was statistically superior to placebo. And in the oral scan, which was the radiographic outcome study, you can again see statistically significant improvement compared to placebo. This is the RA beam. This, again, was a similar trial design to all the other JAK inhibitors, and achieved ACR 20, 50, and 70 response superior to placebo, and also had clinical responses at ACR responses that were statistically superior to adalimumab in this particular study. What we're showing here is the probability plot looking at X-ray progression, standard way that we show progression of radiographic damage. And as with all of the studies with biologics, CSDMARDs, it's about 20 to 25% of patients driving the differential in X-ray outcomes. And again, adalimumab and baricitinib overlapped themselves and had less radiographic progression than the placebo group. And it's clear that these JAK inhibitors do prevent radiographic progression and are disease modifying. The select compare was upadacitinib 15 milligrams plus methotrexate, the approved dose. And what we're looking here is comparison to adalimumab in green and upadacitinib in blue and placebo in orange. And we simplified the slide with left a bunch of pluses and asterisks off this, but upadacitinib was significantly superior to placebo and adalimumab for all measures. And it was also, adalimumab was significantly superior to placebo for all measures. And as you can see on the right part of the slide, at week 26, there was very little radiographic progression with adalimumab as well as upadacitinib compared to placebo. And then this was a fascinating study I happened to be involved with. And this was the SELECT monotherapy in a different design. This was taking patients on methotrexate who had continued active disease. And instead of adding upadacitinib to methotrexate, basically they substituted upadacitinib at the 2 doses for methotrexate. And you can see that upadacitinib monotherapy was statistically superior to placebo, both for ACR20 response and low disease activity by DAS28-CRP. And what was very interesting about this is we've talked for years about suddenly withdrawing methotrexate, flares that you might see in our patients rather than tapering it. These patients had their methotrexate discontinued and were placed on a JAK inhibitor, upadacitinib in this particular example, and did extremely well. And then to show you the rate of response, I think you heard today in the great debate that JAK inhibitors work very quickly. And if you don't see a response, some type of clinical response, by 2 to 4 weeks, you're going to begin to worry. Now, I personally go to 8 weeks with JAK inhibitors. And if a patient has not had a good clinical response at that point, considering moving on, if they've had a moderate response, I'll wait another 4 weeks. And that was seen in this particular study with filgotinib. You can see that by week 4, looking at the 100 and 200 milligram dose, that you already had ACR 20 responses approaching 50%, adalimumab almost in that range. And you can see there was very little difference between the active therapies in this study, filgotinib or adalimumab, compared to placebo over time. The one thing we lack is head-to-head trials. So these are, different populations of patients, heterogeneous. And so you really can't take that to the bank and really make a statement, well, I'm going to pick one JAK inhibitor over another because it was statistically superior. And in some of those cases, statistically superior was 5 to 10 cases out of several hundred. So that's my particular bias on this. Eric or Sheetal, do you have any different viewpoint on that?
No, I would agree. I mean, I think that, the statistical differences are small and, from a clinical perspective, I'm not sure they make a huge difference and don't impact my decision making.
I agree. Clinically, doesn't seem to made a difference thus far.
I mean, the great news is that we have small molecules that are as effective or better than biologics.
Right.
And who would have thunk? And I'm from that era of gold, so I'm that old. So we have gone a very long way from empirical therapies we stole from people to targeted therapies that we have now. And so it's just a wonderful time to be a rheumatologist and have all of these things available to us.
So, I just want to reiterate then. So based on the efficacy data, Stan, that you did a great job kind of taking us through a lot of trials in a very short period of time, do you see a difference between the JAKs when it comes to efficacy at all between the 3 that are commercially approved in this country?
I do not. Based on, again, lacking head-to-head trials, I think the data is very similar. We're going to finish up tonight talking about the very little bit of data that we have about cycling through JAKs. And I think we're all having experience in clinic that we'll see a patient that fails to respond or loses a response to a JAK or has an adverse event to a JAK, and we switch to another one and they do better. But we'll show you a little bit of data that's available and will be presented at this meeting. But I think, there is differences, in the times of day, certain cytokines are being inhibited that might have an impact on one patient and not on another. But we don't have enough understanding of the pathophysiology for a particular patient to really guide us. So at this point, the 40,000 foot level is that, no, I don't think there's any difference.
Okay, and are we aware of any trials coming forward on head-to-head efficacy between the JAK inhibitors?
I'm not aware of them, and I've asked, and people look at me with glazed eyes like, are you crazy? If they're from the pharmaceutical companies, why would we do that trial? So I don't think it'll ever be done by then. I think we're going to learn some information from observational registries, even though they have obviously some indication bias and so forth. But I don't know if we'll get that data. The good news is that Some of the JAK inhibitors will be going generic in about another year in many parts of the world, and the cost will drop, and I think we'll see, maybe then we'll see studies and so forth to inform us. But I don't see anything at this point.
Okay,
I don't see that happening, because I can't imagine who's going to spend the money to fund that sort of trial, because it's the likelihood of showing a meaningful difference is so low. We've never had head-to-head trials between TNF inhibitors because there just was no point. And to do a head-to-head to show that they're equivalent doesn't really get you very far. Perhaps at some point, if we can break down our RA patients to understand either genetically or at the cellular level what makes one patient different from another, then it may make sense to think about head-to-heads in a selected subpopulation, but I don't see it until then.
And now we'll go ahead and turn over on the use of the JAK-STAT MOA in methotrexate naive patients with rheumatoid arthritis.
So Stanley just did a great job talking about the use of these agents in the space that we're largely using them now, in patients who have failed methotrexate, and certainly in patients who failed other biologics when we're looking for another option. But this is the potential promise of JAK inhibitors, of an oral agent. Would this be an appropriate first-line therapy? And as Stanley mentioned, around the world, we may start to see generic JAK inhibitors in the not-too-distant future. It's going to be a few years later than that in the U.S. But when that becomes reality and cost may be more reasonable, would we consider using these agents as first-line? And we actually have trials with each of the 4 drugs that we've been discussing. For tofacitinib, there's the oral START trial, looking at the 2 doses of tofacitinib that were studied in clinical trials. The 5 milligram BID dose is the approved dose, or the 11 milligram XR. Sorry about that. Baricitinib has a trial against methotrexate or combination, using the 4 milligram dose, which is not the approved dose in the US. It's 2 milligrams. Upadacitinib has a monotherapy trial, which we'll talk about, Select Early, using, again, the 2 doses that were studied in clinical trials, the 15 milligram being the dose that's approved in the US. And then finally, filgotinib, which has not yet been approved in the US, has a trial in this population, also at the 2 doses that they studied in their clinical trial program. So we'll start with upadacitinib, and I think this highlights one of the key potential benefits of this approach. This is a trial in methotrexate naive patients who were randomized to 3 different arms, either low dose upadacitinib at 15 milligrams a day, high dose at 30 milligrams a day, or methotrexate with a dose targeting about 20 milligrams a week. There were 2 different co-primary endpoints in the study, ACR 50, so 50% improvement of disease activity, or a DAS28 CRP less than 2.6, which is the DAS28 equivalent of remission of disease activity. And what you see very clearly is both doses of upadacitinib were significantly better than methotrexate from the very beginning, and even followed out to 48 weeks when the sort of slower onset of action of methotrexate might have been expected to catch up if it was equivalent. The difference still remained. On the other hand, with filgotinib, here's a study with filgotinib, either monotherapy at 200 milligrams, the higher dose, or the lower dose or the higher dose with methotrexate, again, compared to methotrexate as monotherapy. And what you see here was not quite as clean, but the combination of methotrexate and filgotinib was more effective than methotrexate at either dose, though statistically at the primary endpoint, the higher dose of filgotinib as monotherapy was not superior to methotrexate. In the tofacitinib trial that I'm not showing you here, tofacitinib was superior to methotrexate. And in the baricitinib trial at the 4 milligram dose, mind you, not the 2 milligram dose that's approved in the US, we don't have data with that dose, it was also superior to methotrexate. So I think all these studies sort of raise the tantalizing possibility of considering moving in this direction as first-line therapy if you have a drug that's superior to what's been our standard, sort of gold standard drug for many, many years. With biologics, this has not been the case with TNF inhibitors. The IL-6 inhibitor antibodies have outperform methotrexate in this population. And now we'll shift gears a little bit and touch on safety. We raised this earlier as a potential differentiator among these drugs, though that hasn't entirely panned out. I think we need to start by thinking sort of broadly about what the various side effects are, the potential side effects of these JAK inhibitors that have been seen in clinical trials. As with any of the agents we use in rheumatoid arthritis, infections tend to be our number one concern. And in particular, a serious infection, which means either hospitalization for infection or requirement for IV antibiotics. Interestingly and importantly for JAK inhibitors, it turns out herpes zoster, a flare of herpes zoster, is a unique infectious adverse event that shows up in clinical trials. And we'll talk about that a little bit more as I show you some of the data. Malignancy is always a concern with any new pathway, any new mechanism of action of drug. But to date, there really has been no evidence that there is an increased risk of malignancy with any of these agents compared to age and gender matched controls. We have heard from Stanley earlier, and it's been an issue that we've been concerned about, is potential hematologic adverse events with these agents. And we do see occasional episodes of anemia, neutropenia, lymphopenia, but for the most part, these have been few and far between. I think not enough to be able to avoid the need to monitor CBCs in patients on JAK inhibitors regularly, but fortunately for the most part have not been a major impediment to therapy. Same thing true for liver function tests. Increased transaminases have been seen, again, in small numbers of patients, again requiring monitoring, but not enough usually to interfere with therapy. Two unique laboratory issues are an increase in serum creatinine and an increase in creatinine phosphokinase, or CK. Mechanism for either of these is not entirely clear. For the creatinine, it may be tubular resorption, but it doesn't seem to be an impact on renal function per se. And the elevated CK doesn't seem to be associated with any clinical muscle abnormalities in these patients. Lipid increases have been seen, and we've been through this before with interleukin-6 inhibitors. No clear evidence that there's an increase in cardiovascular risk associated with increased lipids in these patients, though there's really not a lot of solid data looking at that. We have data with IL-6 inhibitors that has not tied those increases to clinical adverse events. I haven't seen that here, but we don't have solid data there. GI perforations is listed as a question mark, and I think this stems a bit from the experience with IL-6 antibodies and IL-6 inhibitors, where gastrointestinal perforations had been a concern, particularly in patients on steroids or with a history of diverticulitis or inflammatory disease in the colon. Not clearly seen with these agents, but because IL-6 signals through the JAK pathway, that has been raised as a potential adverse event to keep an eye out for. And then finally, and Stanley will talk about this in great more detail in a little bit, venous thromboembolism, which was not really anticipated to be a significant adverse event when trials first started with these agents, but has come up as the trials have moved forward. And so let's look briefly at individual agents. So thinking about the things I just talked about, here's the data from clinical trials with tofacitinib. And what you're seeing here is data comparing the safety profile of tofacitinib with clinical trial data with biologics, either TNF inhibitors or other biologics. And the bottom line is the adverse event profile is pretty much the same. We're kind of used to seeing a risk of serious infections in RA patients on almost all of our therapies of somewhere between 3 and 4 events per 100 patient years. And that's pretty consistent across trials. And the same with tofacitinib. Malignancy rates were not any greater than what was seen with other biologics. Mortality was no greater. MACE events and myocardial infarction was no greater. There was no clear evidence of GI perforation in these trials, but does stand out as on the far right there, herpes zoster, as I mentioned, which is a unique adverse event seen with the JAK inhibitors. Here's the data from baricitinib. And again, serious infection rate, about 3 per 100 patient years, which is in the same range that we typically see with biologics and frankly with methotrexate in historical data. no clear increased risk of any opportunistic infections, no huge increased risk of opportunistic infections, MACE events. Again, herpes zoster seems to be at a somewhat higher risk than we've seen in the past. And I will leave the DVT and PE to Stanley to talk about in a minute. And finally, upadacitinib. Same story, no clear increased risk of malignancies. And here it's compared to the comparator arms in their clinical trials. So either methotrexate or adalimumab as an arm within the same trial. The confidence intervals in most of these are pretty broad. And you really don't see much other than the zoster, once again. And the other thing that does seem to show up here and was fairly evident in the trial data was a somewhat higher risk of serious events in patients on the higher dose, the 30 milligram dose, which was not the dose that was taken forward for approval in the US. And so the 15 milligram dose that was approved and is the dose that's commercially available, but for the zoster, doesn't show a significantly higher risk of any of these other events relative to the comparator treatments. And then speaking of comparator treatments, this is the select choice trial that Stanley mentioned earlier. And interesting here, abatacept, for better or worse, is often considered the sort of least likely biologic agent to cause infections. And here, there was some suggestion that there was a higher risk of serious adverse events and infections with upadacitinib relative to abatacept. The numbers are very small, and it's kind of hard to draw great conclusions from this in a trial of 600 people, 300 in each arm. Small differences in adverse events are a little hard to know how significant they are. And interestingly, in this trial, which is a little bit unexpected, the risk of zoster was the same for abatacept and upadacitinib. But again, small numbers and hard to know what that really means. And then lastly, filgotinib, which, as we know, has not been approved in the US. Here's the pooled safety data for filgotinib. Similar story with some minor differences. And there's a suggestion that perhaps infectious adverse events are a little bit less, and in particular, herpes, which doesn't seem to occur with the same frequency that we've seen with the other JAK inhibitors. I mentioned before that safety differences have shown up and don't seem to be necessarily specificity related, and this would sort of bear that out. This is what is considered a highly specific JAK1 inhibitor, but so is upadacitinib, and there was a higher risk of zoster with upadacitinib. Doesn't seem to be the case with filgotinib in comparison either with adalimumab and methotrexate or methotrexate alone. So it may not be JAK specificity, but there may be something else going on here, if indeed this is the case and pans out with further study. And so with that, I'm going to turn the microphone over to Stanley to focus on a particular safety adverse event that has become something of interest in the last couple of years. Stan.
Thanks, Eric. All right, so we're going to talk just to drill down a little bit on the venous thromboembolism issue with the JAK inhibitors. So this first came to light actually with the FDA review of baricitinib for approval. And this was from the 9 RA clinical trials, 3,500 patients, close to 8,000 patients exposure. And what you see when you look at the incidence rate per 100 patient years, which is on the y-axis, in the placebo-controlled portions of the study, there were 6 cases on 4 milligrams of baricitinib and none on 2 milligrams and none on placebo. So this raised somewhat of an alarm that could baricitinib be associated with the increased risk of venous thromboembolism. When they took a look at extended portions of the trials for both 2 and 4 milligrams and then looked at the totality of the patients' long-term extension, the rates of VTE were similar to what we've seen reported for RA. We know that patients with immune-mediated inflammatory diseases do have higher risk of VTEs. The numbers have varied from meta-analysis 0.3 to 1 per 100 patient years. So there was this signal that was seen in the placebo-controlled portion of the baricitinib trials. Mechanistically, there's no explanation for this, or even with the other JAK inhibitors, tofacitinib. At least baricitinib, there was not the drop in platelet count that we saw, but the patients who had higher platelet counts were not the ones who had DVTs or PE. So this is where the story starts. There have been all kind of analyses done. This is from the phase 3 clinical program, looked at major adverse cardiovascular events, and on the right portion of the slide, VTEs. And what you can see is the event rate was basically no different from adalimumab or patients on methotrexate or placebo for upadacitinib. There's an abstract at this meeting which goes down, it looks at the 35 cases they had in the clinical trials and defines who those people are. And we'll talk about those to a small degree. Same type of depiction for filgotinib, MACE up to 52 weeks. This was FINCH 1, adalimumab controlled, FINCH 3, methotrexate controlled, and this is VT up to 52 weeks. Somewhat busy slides, but you can just scroll across the x-axis and you can see that they had very few events, both for filgotinib, methotrexate, or adalimumab. So did not seem to be a greater number of events there. But then came some data from a FDA-mandated trial, a post-marketing trial, looking at tofacitinib, 5 and 10 milligrams BID, compared to Humira or Enbrel, etanercept, adalimumab, in patients who had higher risk for cardiovascular disease. And this study was powered to look for serious infectious episodes and malignancy, other than cutaneous malignancy. And the patients enrolled were over 50 years of age with moderate to severe RA, typical patients enrolled in trials, but they had to have cardiac risk factors, where it could be hypertension, smoking, previous MI, angina, things of that nature. And what was found while the trial was still ongoing is that in the 10 milligram BID dose of tofacitinib, there was a statistically different rate of pulmonary emboli compared to 5 milligrams BID of tofacitinib and the TNF inhibitors. And there was also a statistically different all-cause mortality with the non-approved tofacitinib 10 milligram BID dose. So this led to this particular cohort of patients being removed from the study or switched over to 5 milligrams BID. There was a numerical trend for DVTs, but that was not statistically significant between the two different doses of tofacitinib or the TNF inhibitors. But based on the findings, the EMA recommended that tofacitinib should be used with caution any patients with known risk factors for VTE, including risk of cardiovascular disease, obesity, cancer, hypercoagulable states, history of prior VTE, as well as patients on hormonal contraceptives, hormone replacement therapy, or undergoing major surgery or immobile. And so very strong recommendation. The FDA's black box warning was not quite as strong, just basically said we shouldn't be using the 10 milligram BID dose that's not approved in RA. And both agencies recommended trying not to use the 10 milligram BID dose for maintenance therapy and ulcerative colitis. We published a paper in the Annals of Rheumatic Disease with Phil Meese, first author, about a couple months ago, and we did an analysis as well of the whole clinical trial program of tofacitinib. And we did identify that not only for tofacitinib, but for patients on comparator, methotrexate or adalimumab, those who had cardiovascular risk had a higher risk of VTE. So that's a given. But when we looked at the totality of the clinical trial program, the rate of VTEs and PEs was much less than in this one particular trial, which led to these black box warnings and these recommendations. So we're still somewhat confused. As you heard Vibeke say earlier and Mike Weinblatt, we don't know the mechanism of action. But I think at this point, in someone who's had a prior VTE, someone who is obese, has higher risk, someone with cardiovascular risk factors, if you have an alternative therapy that you could use, then move on and use a different therapy other than a JAK inhibitor. Again, with upadacitinib and filgotinib from the clinical trial programs, there was really no signal. The patients who developed events were the same patients who had these cardiovascular risk factors, were obese, and so forth. So just keep that in mind. So we have a discussion question here, and I'll go ahead and start it, Sheetal, and let you take over. Based on the safety data, is there a difference between JAKs, and would you select one over another in certain patients? I'll direct the question to you, Eric, first.
Yeah, I don't think so. I mean, there isn't any clear-cut difference. If and when filgotinib is approved in the U.S., you might say, well, perhaps the lower risk of zoster might sway you, but I think the widespread availability and the use of the Shingrix vaccine may sort of mitigate that, and that may not be an issue at all. So I really haven't seen much that would make me choose different agents through any of this.
And, I think this has been a hot area of debate and talk kind of throughout the day ever since the great debate. But would you use a JAK inhibitor in a patient that's had a past medical history of a venous thromboembolism? So let's say they've had a history, and let's say they're actually on anticoagulation. At that point, would you consider actually using a JAK mechanism of action on them? Eric or Stan?
Well, I was going to say, I've faced this same challenge with a patient of mine who'd had adverse events to pretty much everything else I had to give him, and had a PE, spent a lot of time discussing with the hematologist. He's on ongoing anticoagulation, and with the recognition that we need to do something to control his disease and the patient involved, we're going to keep going, but we'll see how it goes. I mean, it's not a preference. It's not something I would like to do, but it's certainly something that I've found myself having to do in at least one situation.
And Eric, you said this patient was on chronic anticoagulation, or?
That's correct. Yeah.
So I had the luxury of being in a group of about 10 to 15 people, which some were rheumatologists, but the rest were clot people. And they were a little bit interesting, a little cavalier, but they felt that if someone was on anticoagulation, no problem. Now, can they quote me data? No. Or science? No. But I can just tell you that this was sort of a RAND UCLA method of going through this. And the clot people, of course, if they're on anticoagulation, we don't have concerns. Yeah. But for me, if someone's had a past history of VTE or PE, I would be very reluctant unless I had no other options for the patient. The bigger struggle is, some of the sub-analysis is suggesting that oral contraceptives may increase the risk. Some of the sub-analysis have not. For example, Ernie Choi's presentation at this meeting, they did not find that oral contraceptives, very small numbers of patients, or ERT increased the risk. So it's really for the other group of people. If someone who's had a VTE or PE, then I think that I think we should avoid these at this point.
Yeah.
So given the known risk, you should avoid it if you can, but we're in a data-free zone when it comes to using it in patients on chronic anticoagulation.
Yeah. And I think it's also a data-free zone on who's truly at risk. I mean, I think that's one of the challenges we face is the labeling says if they're at high risk, but RA patients have a high risk of VTE, period. And so are they just at higher risk and excluded altogether? I mean, you think about obese patients, certainly a patient who's had a history, there's no question about it. But, right. And I'm not dismissing that as an issue, but it's a struggle. It's like, if we're going to use that as a decision point, then what are the factors that we use to conclude that someone is truly high risk and is not an appropriate patient? It's a tough one.
No, this is a real tough one. And it was brought up a lot of questions this morning at the great debate as well. So thank you. I'm going to go ahead and move on now. You've heard a great amount of information that's already been presented to you regarding the data and of safety and efficacy in these patients. And now Dr. Cohen's going to go ahead and tell us in the clinic, what do we know when it comes to using JAK inhibitors? Go ahead, Stan.
So the question has been from the beginning, and since we're not going to have switch studies, head-to-head studies, if you have a patient who fails one JAK inhibitor, will they respond to the next? So this paper was actually presented at EULAR and is being presented at this meeting. Small study, basically a clinic's experience, 31 patients, half had received baricitinib first, half had received tofacitinib first. The patient characteristics are here. They had failed multiple previous biologic DMARDs. 87% of patients have been on prior biologic DMARDs. 2/3 were ACPA positive, and they had significant disease activity. This is the data. And what they showed is that regardless of which one they were on first and switching, patients improved. And this was a median follow-up of 19.5 months. They had reduction in the steroid dose, improvement in the DAS CRP, not CPR. And so again, I think this is what I'm seeing in the clinic, and when we have the discussion, we'll see what Sheetal and Eric also see. But clearly there are patients who may have a less response to one JAK inhibitor, will respond to another, and I look forward to much larger observational registry data to confirm that. Now, our last slide on the clinical experience, This was a study I was involved with as well, and some of you may have been investigators. And the question was, in patients on combination therapy with methotrexate who are in low disease activity, as measured by CDAI, if you withdraw methotrexate, how will the patients do over the next 24 months? And in this particular study, it was a non-inferiority design. We showed that sudden withdrawal of methotrexate in a blinded fashion had very little impact on clinical response in patients who had been in low disease activity for 6 months. So we know our patients don't like taking methotrexate, many of them. We know they don't like taking multiple medications. They vote with their feet. They also come in and you ask them what the dose of methotrexate they're on, and they tell you, well, I stopped that 4 months ago. So this study at least confirms that for tofacitinib, similar to what we saw with upadacitinib, that you can discontinue methotrexate and patients may do well. So again, I think this is similar to what we're seeing in clinic, where about 30 to 40% of our patients on JAK inhibitors are on monotherapy. So what's the place of JAK inhibitors in RA treatment? We have the EULAR guidelines, and on Monday, we will have the new ACR pharmacologic recommendations. And we'll remain to see where the targeted synthetic DMARD JAK inhibitors are placed. What will happen in 5 years? Eric, you want to give your thoughts on that?
Well, I mean, I think that's where the cost issue comes in. In the U.S., at least, it sounds like tofacitinib goes off patent, I believe, at the end of 2025, or it's either 24 or 25, I don't recall. But I mean, that's the number, and we haven't seen a huge influx of biosimilars in the U.S., but that's because of the sort of unique structure of the way things are paid for in our country. But it may change with true generic JAK inhibitors. And when that starts to happen, if a generic tofacitinib is significantly less expensive than a biologic, it may be the way to go in that population and suddenly open up the option as first-line therapy if it's truly cheap enough. So we'll see.
We're in our clinic, we have a clinical trial center, and we're working with a lot of early things in phase 1. And they start talking about what's going to be the active comparator in a study moving forward. And we said, you better be an active comparator to a JAK inhibitor that's going to be generic. Yeah. If you want to have any utilization of your product in 5 years. So I think that's what we're going to. And then I'll address the comorbidities, and I'll shut up and let Sheetal take us home. I think of JAK inhibitors somewhat similarly to a drug I worked with a great deal in development many years ago, leflunomide. I think they're fantastic drugs, but I think they have a very narrow therapeutic and toxicity window. I think we've got the right doses. We got rid of some of the doses that were too toxic in the clinical trials, which is what clinical trials are for. You have to be careful in patients with multiple comorbidities. We know that with all of our therapy, 65 and older is a high-risk population. But I think patients with COPD, recurrent infections, these would not be my first choice.
Thank you, Dr. Cohen. To hear the next episode in this series, click on the link in the show notes.